Welcome to this in-depth exploration of a groundbreaking scientific discovery that challenges what we thought we knew about fetal protection. For decades, the placenta was considered an almost impenetrable fortress guarding developing babies from harmful substances. But a recent study has revealed something deeply concerning. Nanoplastics, plastic particles smaller than a micrometer, are crossing this barrier and reaching developing fetuses. Today, we'll dive into this remarkable research, understand its implications, and explore what this means for human health and our relationship with plastics. Last month, researchers from the University of New Mexico and Utrecht University published a landmark study in the Environmental Science and Technology journal. Using advanced mass spectrometry and fluorescence microscopy, they found comprehensive evidence that nanoplastics can cross the human placental barrier. The team identified an average of 17 distinct types of plastic particles in placental tissues collected from 62 volunteer participants who had recently given birth. This discovery marks the first definitive proof that these tiny plastic particles can infiltrate what was long considered one of the body's most secure biological barriers. As the study's lead author, Dr. Amina Rodriguez explains, the placenta has long been considered a nearly impenetrable fortress protecting developing fetuses. Finding nanoplastics not just in the placenta, but in the umbilical cord, blood, and fetal tissues challenges that assumption entirely. What makes this finding particularly alarming is the size of these particles. While previous research had identified microplastics in human blood and organs, this study specifically focused on nanoplastics, particles smaller than 1 micrometer, which is about 1/70th the width of a human hair. At this microscopic scale, these particles can potentially interact with cellular mechanisms and even penetrate cell membranes. To appreciate the significance of this discovery, we need to understand what the placenta actually does. Far from being a simple barrier, the human placenta is a sophisticated regulatory interface between mother and developing child. It controls the transfer of nutrients from mother to fetus, facilitates gas exchange providing oxygen while removing carbon dioxide, eliminates waste products, and provides crucial immunological protection. The research team observed something concerning in their samples. Tissues with higher nanoplastic concentrations showed subtle but measurable alterations in placental architecture. These changes were particularly notable in the syncytiotrophoblasts layer, the specialized tissue layer that directly interfaces with maternal blood. This layer serves as the primary site for nutrient and gas exchange, and any disruption to its structure could potentially impact these vital functions. The placenta has evolved sophisticated mechanisms to selectively allow beneficial substances to pass through while blocking harmful ones. Finding that nanoplastics can breach this defense system raises profound questions about what other environmental contaminants might similarly bypass this critical protection. Perhaps the most concerning aspect of this research isn't just the presence of nanoplastics themselves, but what they bring along with them. The study revealed that these plastic particles had accumulated various compounds during their environmental journey before reaching human tissues. These hitchhiking substances included heavy metals like lead, mercury, and cadmium, persistent organic pollutants including PCBs and flame retardants, plasticizers and other plastic additives such as phthalate and bisphenols, and even antibiotic-resistant microbial biofilms that had colonized the particles' surfaces. As environmental toxicologist Dr. Wei Zhang noted, these particles essentially function as molecular Trojan horses, potentially delivering harmful compounds directly to developing fetal tissues that would otherwise be protected. The research revealed a particularly troubling mechanism at work. The nanoplastic's high surface-to-volume ratio creates an ideal platform for the absorption of environmental contaminants. Laboratory analyses showed that a single nanoplastic particle could carry up to 30,000 molecules of adsorbed compounds, effectively concentrating toxicants and delivering them across biological barriers. Furthermore, the team discovered that the placental environment, rich in proteins, lipids, and various biological molecules, enhanced certain plastic types' binding capacity, particularly polystyrene and PVC derivatives. The researchers identified several types of polymers in the placental samples. The most commonly detected were polyethylene, PE, polypropylene, PP, and polyethylene terephthalate, PET, all of which are ubiquitous in our daily lives. Polyethylene is found in plastic bags and food packaging. Polypropylene appears in food containers, bottle caps, and textiles. Polyethylene terephthalate is common in water bottles and food containers. Spectroscopic analysis revealed something particularly concerning about these particles. Many had undergone surface modifications during their environmental transport, developing charged regions that attracted polar contaminants. Additionally, the particles had acquired what scientists call protein coronas, coatings of proteins that potentially disguise them from immune recognition. This biological cloaking might explain how these foreign particles evade maternal-fetal immune surveillance mechanisms designed to prevent non-self materials from reaching the fetus. Essentially, the nanoplastics may be wearing biological disguises that trick the body's defense systems into treating them as natural substances rather than foreign invaders. This would explain how they successfully navigate past immune checkpoints that would normally identify and eliminate foreign particles before they could reach the developing fetus. The research team didn't just identify the presence of nanoplastics, they conducted parallel studies on exposure pathways by analyzing participants' dietary habits, occupational exposures, and living environments. Their findings suggest multiple routes through which these particles enter maternal circulation. Inhalation appears to be a primary pathway. Indoor air contains surprisingly high concentrations of airborne nanoplastics, particularly in environments with synthetic carpeting, furnishings, and poor ventilation. One participant who worked in a textile factory showed nanoplastic concentrations nearly 3 times higher than the group average. Dietary exposure was another significant factor. Participants who regularly consumed food and beverages from plastic containers had elevated levels of certain polymer types. The correlation between bottled water consumption and higher PET nanoplastic concentrations was particularly notable. The researchers also found that cosmetics and personal care products containing microbeads or plastic-based ingredients contributed to exposure, potentially absorbing particles through the skin or mucous membranes. The team observed significant seasonal and geographical variations in exposure patterns. Urban participants exhibited nanoplastic profiles dominated by tire wear particles and synthetic textile fibers, while rural participants showed higher proportions of agricultural plastic degradation products. Winter samples contained elevated levels of acrylic and polyester particles, correlating with increased use of synthetic clothing and reduced ventilation in heated indoor environments. To better understand exposure sources, the research team developed a novel methodology they called the Plastic Exposure Index. By combining biomonitoring data with detailed questionnaires about lifestyle factors, they created a comprehensive picture of how these particles enter our bodies. This approach revealed unexpected exposure sources that many of us encounter daily: dental floss, tea bags, disposable face masks, and certain medications with polymer-based coatings or delivery systems. The index also revealed potential mitigation strategies. Participants using menstrual cups or period underwear as alternatives to plastic-containing feminine hygiene products showed significantly lower levels of certain polymer types. This suggests that conscious consumer choices might help reduce exposure to some nanoplastics. The Plastic Exposure Index represents a new approach to understanding our complex relationship with plastic materials. Rather than focusing on single products or behaviors, it examines the cumulative effect of multiple exposure pathways. This holistic approach helps identify which interventions might be most effective in reducing overall nanoplastic burden in the body. The researchers hope this index will evolve into a practical tool that individuals can use to assess and reduce their own exposure risks, particularly during sensitive periods like pregnancy. While the study doesn't definitively establish health consequences, it opens urgent new questions about developmental impacts. The research team has begun a longitudinal follow-up study tracking the children born to study participants, monitoring developmental markers, immune function, and metabolic parameters. Concurrently, laboratory studies using placental organoids and animal models are investigating mechanisms by which these particles might affect fetal development. Preliminary results suggest potential disruption of placental nutrient transport and hormone production. The implications extend beyond pregnancy. Policy experts call this a watershed moment for regulatory approaches to plastic pollution. As environmental policy analyst Dr. Hiroshi Tanaka notes, we've been focused on environmental impacts of plastics for decades. This research shifts the conversation toward direct human health impacts, particularly for the most vulnerable, developing fetuses. The research team is also developing new analytical methods to detect even smaller plastic particles. Current limitations in detection technology mean we may be seeing only part of the problem. As Dr. Rodriguez puts it, what we're measuring may just be the tip of the iceberg. The potential long-term effects of nanoplastic exposure during fetal development remain largely unknown. but the presence of these particles in developing tissues raises concerns about possible impacts on organ development, immune system function, and even neurological development. The study has catalyzed an unprecedented multidisciplinary collaboration between materials scientists, obstetricians, toxicologists, and environmental engineers seeking immediate and long-term interventions. A consortium of research institutions has launched the Maternal Environmental Protection Initiative, MEPI, focused on developing practical guidelines for reducing nanoplastic exposure during pregnancy. Immediate recommendations from this initiative include filtering drinking water, minimizing food contact with plastic packaging, improving indoor air filtration, and reevaluating materials used in prenatal supplements and medical devices. More ambitious efforts include engineering biodegradable polymers that minimize nanoplastic generation during breakdown. Dr. Eliza Montgomery, a reproductive endocrinologist collaborating on the follow-up studies, emphasizes that the findings shouldn't cause undue alarm. While these results demand serious attention, it's important to note that we're still determining clinical significance. Pregnant women should focus on reasonable precautions rather than anxiety-inducing measures. This balanced approach acknowledges the concerns while emphasizing that research is still evolving on the exact health implications. The collaboration also highlights how complex environmental health challenges require expertise from multiple scientific disciplines working together to develop comprehensive solutions. The discovery has accelerated innovation in detection technologies. A team at Stanford University is developing a smartphone-compatible sensor that could allow consumers to test products for nanoplastic release potential. This device would use specialized optical sensors to detect the characteristic signatures of various polymer types, potentially allowing individuals to make more informed choices about the products they use. Meanwhile, environmental monitoring stations are being upgraded to track nanoplastic concentrations in air and water. These next-generation monitoring systems combine advanced filtration techniques with automated spectroscopic analysis to provide real-time data on nanoplastic pollution levels. This information could help identify pollution hotspots and evaluate the effectiveness of mitigation strategies. The technological response extends to remediation approaches as well. Materials scientists are developing advanced filtration materials specifically designed to capture nanoplastic particles from drinking water and indoor air. Some of these materials use biomimetic approaches, inspired by how biological systems filter particles, while others employ novel nanomaterials with precisely engineered surface properties that selectively bind to common polymer types. These technological innovations represent a rapid response from the scientific community to address both detection and mitigation of nanoplastic exposure. Perhaps most significantly, the research has sparked renewed interest in the precautionary principle in chemical regulation. Several European nations have announced plans to revise risk assessment protocols for consumer products, incorporating nanoplastic leaching potential as a standard evaluation criterion. This represents a fundamental shift in how we regulate plastics, moving from focusing primarily on their intended use to considering their entire life cycle, including degradation products. The regulatory response varies globally. While European countries are taking a precautionary approach, implementing stricter standards before all health effects are fully understood, other regions are waiting for more definitive evidence of harm before making significant regulatory changes. This creates a natural experiment in different regulatory philosophies. Industry response has also been mixed. Some companies are proactively reformulating products to reduce nanoplastic generation, seeing this as both an ethical imperative and a market opportunity. Others are waiting for regulatory requirements before making significant changes. Consumer advocacy groups are pushing for greater transparency, demanding clear labeling of products containing plastics that may generate nanoplastics during use or disposal. This regulatory awakening reflects a broader reconsideration of how we manage the potential health impacts of materials that have become ubiquitous in modern life. While research continues, the study authors have outlined several practical steps individuals can take to reduce potential nanoplastic exposure, particularly during pregnancy. For drinking water, they recommend using solid carbon block filters certified to remove particles at the nanoscale and avoiding bottled water in plastic containers when possible. For food storage, they suggest replacing plastic containers with glass, stainless steel, or ceramic alternatives, particularly for hot foods and liquids, which can accelerate the leaching of particles from plastic containers. For indoor air quality, regular ventilation and HEPA air filtration can reduce airborne nanoplastic concentrations, especially in spaces with significant synthetic materials. With personal care products, the researchers advise checking ingredients for microbeads and plastic-based components, choosing natural alternatives when available. For clothing and home textiles, natural fibers shed fewer synthetic particles than polyester, acrylic, and nylon materials. The researchers emphasize that these precautions represent reasonable steps based on current knowledge, not cause for alarm. They also note that individual actions, while important, need to be complemented by broader systemic changes in how we produce, use, and dispose of plastic materials. The goal isn't to eliminate all plastics, many of which serve important functions, but to develop a more thoughtful, precautionary approach to their use, particularly for applications that might lead to human exposure. This groundbreaking study opens numerous avenues for future research. The research team has outlined several priority areas for continued investigation. First, they're working to develop even more sensitive detection methods that can identify smaller plastic particles and determine whether they penetrate not just into fetal tissues, but into individual cells and potentially even cell nuclei where they might interact with genetic material. Second, longitudinal studies are being established to track potential health outcomes in children exposed to nanoplastics during fetal development. These studies will monitor everything from developmental milestones to immune function, metabolic health, and even neurocognitive development. Third, laboratory studies using advanced cell culture systems and animal models are investigating the mechanisms by which nanoplastics might influence cellular function, with particular attention to potential endocrine disrupting effects and impacts on placental transport functions. Fourth, environmental scientists are mapping nanoplastic distribution patterns to identify exposure hotspots in vulnerable populations. This includes developing predictive models for how nanoplastics move through ecosystems and eventually reach human tissues. Finally, materials scientists and chemical engineers are researching alternative polymers that maintain the beneficial properties of plastics while minimizing the generation of persistent nanoplastic particles. This includes both truly biodegradable materials and polymers designed to break down into non-toxic components rather than persistent particles. The discovery of nanoplastics crossing the placental barrier represents a pivotal moment in our understanding of environmental health. What we once considered a secure biological fortress protecting our most vulnerable, developing fetuses, has now been shown to be permeable to some of the smallest byproducts of our plastic-dependent world. These invisible invaders not only breach this critical barrier but potentially bring along a cargo of environmental contaminants, functioning as molecular Trojan horses delivering toxicants directly to developing tissues. As we've explored throughout this video, the implications extend across multiple domains, from reproductive health to materials science, from environmental monitoring to regulatory policy. While this discovery raises legitimate concerns, it also catalyzes important innovations in detection technology, mitigation strategies, and alternative materials development. The multidisciplinary response demonstrates science at its best, rapidly mobilizing diverse expertise to address an emerging challenge. For individuals, particularly expectant mothers, this research offers actionable insights for reducing potential exposure while avoiding undue alarm. For society, it prompts a reevaluation of our relationship with plastic materials, moving toward more thoughtful application of the precautionary principle. As research continues to unfold, one thing is clear. Our understanding of environmental health must encompass not just the visible world, but also the invisible particles that increasingly define our modern environment. The story of nanoplastics crossing the placental barrier isn't just about pollution. It's about reconsidering the boundaries between our technological world and our biological selves. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.